I ended my previous video in this slide and I will be proceeding further on from here. So coming to the quiescent center. Now in between the root cap and the meristematic region of the root, there is a region containing inactive cells and that region is called the quiescent center. Now it was first discovered by Clove in maize. The region is made up of about thousands of inactive cells. The rate of cell division of the cells is very slow as compared to the surrounding cells, as the cells are mostly in the G0 phase. Now, the amount of DNA, RNA and protein is also less in this region. Now basically this region acts as a reservoir of meristome. That is, when the root tip is damaged, this zone restores the growth by providing the meristematic cells. This is one of the most important slide and one of the most important portion for your NEET questions. Questions come from this section, that who discovered this quiescent center, what is quiescent center, what is the stage of cell division of the cells in this quiescent center, that is a G0 stage. So these are the few questions which actively comes in your NEET exam. Now coming to the permanent tissues. Now as I already told you that the cells of the permanent tissue does not divide frequently, and that is why the name permanent. They are formed by the division and differentiation of the meristematic tissues, that is the primary meristem. A permanent tissue can be divided into two types, the simple permanent tissue and the complex permanent tissue. Again, the simple permanent tissue can be of three types, that is the parenchyma, collenchyma and sclerenchyma, whereas the complex permanent tissue can be of two types, the xylem and the phloem. Now, the simple permanent tissue is called simple permanent tissue because it is made up of cells which are actually similar in structure and function, whereas the complex permanent tissue is called so because it is made up of different types of cell. Now coming to the first of the simple permanent tissue, that is parenchyma. So it is a primitive tissue and it is the first evolved tissue. Now remaining all tissues are derived from this tissue, so it is also called the fundamental tissue. Now the cells are of various shapes, that is barrel shaped, spherical or polygonal. The cell wall is made up of cellulose. The cells are isodimetric. The intracellular space is present. Each cell contains a large central vacuole for the storage of food, and that is the main function of these parenchymatous cells, that is the storage of food and nutrients. Now there are different modifications of this parenchyma tissue found in the plants, and these modifications are parenchyma, prosenchyma, chloroenchyma, idioblast and mucilage parenchyma, to add a few. Now coming to the first one, that is the parenchyma. Now, as the name suggests, this parenchyma has large air spaces. It is found in hydrophyte and it gives buoyancy to the plant, which helps the plant organs to float. Then there is chloroenchyma. The word chloro signifies chloroplast. So the parenchyma with large amount of chloroplast is called the chloroenchyma, and it is found in mesophyll of leaves and helps in photosynthesis. Now coming to the prosenchyma, the prosenchymatous cells are long and tapered at both ends without any intracellular space, and it helps in mechanical support. The idioblast are parenchyma that stores unwanted substances like tannins, resins, etc. Whereas then there is another type of parenchyma called the mucilage parenchyma, in which large vacuoles are found which stores water, as in case of aloe vera. Now coming to collenchyma, another simple permanent tissue. The cells of the cell wall of this type, these cells of this tissue are made up of cellulose, that is by living. The cell can be of various shapes, oval, elongated, spherical, etc. It consists of cells which are much thickened at the center, and this is, remember that this is one of the prime feature of collenchyma, that is the cell wall thickening. This is very much important, and this thickening is basically due to the deposition of cellulose and hemicellulose. The intracellular space is absent, and the main function of chloroenchyma is to assist in photosynthesis, as it also contains chloroplast, and in addition to it, they provide mechanical strength to various growing plant organs. But remember, the providing the mechanical strength is not the basic function or the main function of this chloroenchyma. Now collenchyma can be of three different types. One is the angular collenchyma, One is the lamellar parenchyma and another one is the tubular parenchyma. So angular, in case of angular parenchymas, the deposition is actually at the corners of the cells. You can see that the depositions are at the corners of the cells. But in case of lamellar parenchyma, the depositions are along the radial walls. The plate-like depositions are found in the tangential walls. Whereas in case of tubular parenchyma, the depositions are all around the cells. So these are the three different types of parenchyma on the basis of cell wall thickening. Now coming to sclerenchyma. Sclerenchyma are the main mechanical tissues, and the cells are thick-walled, lignified and dead. Now remember one thing always, that the cells in which the cell wall contains lignin are dead, because lignin is a hygroscopic substance and it does not allow water to enter into a cell, as a result of which the cells dies. So whenever you will see that a cell contains lignin, remember that it will be a dead cell. And the main function of sclerenchyma is mechanical tissue. So this is very important, that if question comes that which is the main mechanical tissue of a plant, so always take sclerenchyma, not chlorenchyma. The sclerenchyma can again be classified into two types, that is the sclerids and the sclerenchyma fibers. Now the sclerids can be of 5 different types, and the sclerenchymatous fiber can again be divided into the xylary fiber and the extraxylary fiber. So let us first discuss sclerenchyma fiber. Now these are very much elongated cell. As you can see, these are elongated cells with tapering and pointed ends. The ends are pointed. Wall is made up of lignin, and the fibers. And remember one thing, that these fibers are one of the longest cells in the plant bodies. According to position, there are two types of this sclerenchymatous fiber. One is the xylary fiber or wood fiber, and another one is the extraxylary fiber. There is a print typing mistake. This will be xylary fiber. Xylary fiber or wood fiber is basically the fibers associated with xylem, as the name suggests, and these are not flexible type of fiber. Whereas the extraxylary fibers are fibers which are present in any other part of the plant except xylem. That is why the name extraxylary. And these are the one of the most flexible fibers, or the flexible fibers of the plant body. So these are basically the commercially important fibers. For example, phloem fiber, cortical fiber, that is the bast fiber, the hemp, which is the hemp, jute, flex. These are all the type of extraxylary fibers. Now coming to the sclerids. Now they have extremely thick wall. The lignin deposition is very much. The lumen is narrow, and they are mostly found in the hard parts of plant and provide rigidity. Now, according to shape, the sclerid can be of 5 different types. One is the macrosclerid, that is the rod shaped. It is large and rod shaped, and it is found in hard seed coat of members of Fabaceae family. Another is the brachysclerid, or which are also called the stone cell. It is small, oval or rounded, and it is found in cortex, pith, and phloem. Whereas then there is the trichosclerid. This is the trichosclerid. This is hair-like and is found in the epidermis, which actually helps in the formation of stem hairs or all. And then there is the astrosclerid, which is the star-shaped sclerid. It is found in the intracellular spaces of leaf and stems of hydrophytes. And then lastly there is the osteosclerid. As the name suggests, it is bone-like, and it is found in the hypodermal layers of seed and fruit. So these are the different types of sclerenchyma. Now coming to the complex permanent tissue. So now these tissues are called complex as they are composed of different types of cell. It is of two types. One is the xylem and another one is the phloem. And the xylem and phloem together constitute the vascular bundle. Firstly, coming to the xylem. It functions as a conducting tissue for water and mineral, as you all know. It conducts water and minerals from the root to the stem and the leaf, and it also provides mechanical support to plant. Now, on the basis of origin, xylem can be of two types, the primary xylem and the secondary xylem. Now, the primary xylem is originated from procambium, whereas the secondary xylem is originated from vascular cambium. Now, uh, The vascular cambium is a type of secondary meristem, and on the basis of shape or the primary xylem can again be divided into two parts, the protoxylem and the metaxylem. The first formed primary xylem element is called the protoxylem, that is by the name pro means primitive, whereas the later formed primary xylem is called the metaxylem, and the cells of protoxylem are small as compared to metaxylem, whereas the cells of secondary xylems are isodiametric, that is there is no differentiation in the size of the cells of a secondary xylem. In case of stem, the protoxylem lies in the center and the metaxylem towards the periphery, and this type of primary xylem arrangement is called enlarged condition because the protoxylem is toward the center, whereas in case of roots the protoxylem are toward the periphery and it is called exarch condition, whereas in case of leaves the protoxylem is surrounded by metaxylem and this condition is called visarch, as the protoxylem is surrounded by two metaxylem. Now, the xylem can be divided into four parts: the tracheids, the vessels or the trachea, the fibers, and the xylem parenchyma. Of these three parts, that is the tracheids, the vessels or trachea, and the xylem fibers, are the dead parts of the xylem. The xylem parenchyma is the only living part of the xylem. Now, you can see that why the xylem is called the complex permanent tissue, because it is made up of both dead and living types of cells. Now, as the maximum part of the xylem or the maximum constituent of a xylem is made up of dead part, so that is why it is grossly called as a dead tissue. Now, if we see the if we come across the evolutionary aspect of xylem development, then we will see that in case of pteridophytes, no trachea is found, just with a single exception in Selaginella, where a trachea or a vessel is found. Same is the case with gymnosperm, with the exception of Netum only, where the trachea or the vessels are found. In case of angiosperms only, the trachea or the vessels are found. But there are certain exceptions of angiosperms also, where the vessels or the tracheas are absent, and those exceptions are Trococentron, Tropodendrin, Yucca, Dracaena. These are certain angiosperms which lacks vessel. Now, let us look into each and every component of the xylem. So, coming to the first one, that is the tracheids. These are elongated, narrow lumen cells with tapering ends. The cell wall is made up of lignin, that is by dead, and the cells are long and placed one upon the other. Coming to the vessels or the trachea, these are elongated, cylindrical with wider lumen. The walls are lignified, the end walls is perforated. The vessels and trachea forms the main conducting tissues of the plant, and these are also called together as head row. Now, the vessels are example of dead syncyte. Now, remember the term syncyte. The syncyte is basically a type of cell which is formed by the joining of many different cells. This is called a syncyte. Now, as the vessels are dead cells, so that is why it is an example of dead syncyte. Now, coming to the xylem fiber, as I already told you that these are the sclerenchymatous fiber, the xylary sclerenchymatous fiber, thus dead, and they mainly provide the mechanical support to vessels. Whereas the xylem parenchyma are the only living cells of the xylem. They store food matter and they help in the radial conduction of water. Now, coming to phloem. Now, the phloems are also called bast or lepton, and remember one thing, xylem is also called wood, whereas the phloem is called the bast or lepton. Now, on the basis of origin, phloem can also be of two types. One is the primary phloem, which is derived from procambium, which is a type of primary meristem, whereas the secondary phloem, which is formed from vascular cambium, which is a type of secondary meristem. Now, primary phloem can again be divided into two types, that is the protophlloem and the metaphlloem. The first formed primary phloem consists of narrow sieve tube and is called the protophlloem, and the later formed primary phloem has bigger sieve tube and is referred to as the metaphlloem. Remember that there is no size demarcation in case of metaphlloem, protophlloem and metaphlloem, as it was in the case with protoxylem and metaxylem. Now, there are four different components of phloem: the sieve tube elements, the companion cells, the phloem parenchyma, and the phloem fiber. Of this, the sieve tube elements, the companion cells, the phloem parenchyma are the living part of the phloem. Just phloem fiber is the only single dead part of the phloem, and as the phloem consists of more living parts, so grossly the phloem is also called as a living component or a living tissue. Now again, if we look into the evolutionary aspect of this phloem development, so we will see that in case of pteridophytes, sieve tube is not present, a sieve cell is being found. Sieve tube is basically the is formed by the continuation of the sieve cells. The sieve cells join together to form the sieve tube, which is only found in case of the angiosperms. In case of pteridophytes, only sieve cell is found and no companion cell is found. Similar in case of gymnosperm, also sieve cell is found, no sieve tube. Phloem fiber is found, phloem parenchyma is found, and no companion cell, but a substitute of companion cell is found in case of gymnosperm, and that is called the albuminous cell. Only in case of angiosperm, sieve tube is found as well as the companion cell. The phloem, one important thing is that in monocots, phloem parenchyma is absent with a single exception of ranunculus. Ranunculus is a monocot which contains phloem parenchyma. Now, if we look into each and every component of this phloem, you will see that they are coming to the sieve tube. They are long tube-like structure and are associated with companion cells. Their end walls are perforated and is called the sieve plates. Just remember that if you have to form a tube from, then different cells must combine one upon the other. So there must be some connection from each and every cell so that the tube can function, and that is why the end wall of each and every cell is perforated, and the end walls of each cell is called the sieve plates. Now, callose, which is a complex carbohydrate and its chemical formula is beta-1,3-glucan, deposits on the radius of these sieve pores during autumn to form a thick layer. This is called a callus pad. It prevents, it serves as a two function basically during the senation period or during autumn, during autumn when the leaves set. So basically this callus actually prevents the movement of food matter to those old parts so that the food matters can be used by the new parts of the cells. And this callus pad actually serves another function, that is prevent the spread of it prevent the plant from bacterial infection. That whenever a bacteria or a virus infect a plant, it basically travels from one part of the plant to another by the help of these vascular bundles and mostly through this sieve pores. So basically callus deposition prevent the spread of this virus or bacteria or any microbe from one part of the plant to another. So this callus pad performs two functions. Now a mature sieve tube or sieve cell lacks nucleus and it is an example of living syncyte. Remember I told you the vessels or the trachea are the examples of dead syncyte. Similarly these sieve tubes are an example of living syncyte. And one more thing, the sieve tube lacks nucleus. Now coming to the companion cell, the functions of sieve tubes are controlled by the nucleus of companion cells. And since sieve tube elements and companion cell originate together, so these are called the sister cells. Now these two cells are connected by plasmodesmata so that the cytoplasmic connection is maintained. Coming to the phloem parenchyma, it is living, it is also known as the vast parenchyma, and as you all know that as the main function of parenchyma, the function of phloem parenchyma is also storage of food, and it is absent in monocot with an exception of ranunculus. Now coming to the phloem fiber, it is a type of sclerenchymatous fiber, as I already told you, extraxillary fiber. It is the only dead portion of the phloem and it is also known as the bast fiber. So with this I completed or I came to the end of the different types of tissues in plant, and I will be proceeding to the different tissue systems in the next video of this chapter.